Okay.
Start now.
Hi, I'm Vishal, and me and my groupmates gonna explain about polymerase chain reaction. Polymerase chain reaction is a method used by biologists, specifically molecular biologists, to make millions to billions of copies of a specific DNA sample in a small amount of time. This process comes with three different stages. First is denature, second is annealing, and third is extension. Okay. For this process, we need several tools. The first tool is the first material is the sample of DNA. The particular sample of DNA will be replicated in this process. And then we need a primer. Primer need in a large quantity where primer gives a starting point for the polymerase to continue the replication process. And then we need tag polymerase. Tag polymerase is a polymerase taken out from a particular bacteria which called Thermus aquaticus, where it has a high heat stand for the denature process. And then we need nucleotides. Nucleotides is basically to continued by the tag polymerase to complete the double strand. And then we need test tube to conduct this entire experiment, and buffer solution which contains KCl and 3HCl. That's all. Thank you.
Okay, thank you, Vishal, for the announcement, for your previous presentation. Okay, now it's my turn, and I'll be explaining for the steps for PCR. Okay, so as said before, there are a few needed materials that we need for this process. First, we need the template DNA, the tag polymerase, the primers, and the nucleotides. So basically, what we're going to do is to, as I said before, make a bunch of copies of the specific DNA. So the first step is basically you take a test tube, take the isolated DNA and mix them with primer. Then we go into the very first step, which is the denaturing. The denaturing process involves heating up the isolated DNA and primers up to 90 degrees Celsius for 15 to 30 seconds. This causes the DNA double helix to separate and allow to basically denaturing them and allowing them to anneal for the next process, which is the next step. It's called annealing. Annealing is where the primers are bind to the DNA sequence and initiate polymerization. This can only occur once when the temperature is lowered to around 60 degrees Celsius for 20 to 40 seconds. One primer is bind to each strand. So for the third and last step, which is the extension step, is basically the replication of the new specific DNA. DNA polymerase, which is the tag polymerase enzyme, will join free DNA nucleotides and bind them together to form the new DNA strands. So basically, you will get two specific DNA strands, which is the original strand and the new DNA strand. Each of this process is repeated around to 15 to 30 times. That is all for me. So you can go on to the next person.
Thank you, Amin, for your presentation. Now, my name is Raul, and now I'll be talking about the advantages and the applications of PCR. So, PCR has a number of advantages. One of the many advantages is that it is fairly simple to use and to understand and produces rapid results. The technique is highly sensitive and it is able to produce millions, if not billions of copies of specific products for sequencing, cloning, and analysis. PCR is a very powerful and practical research tool. And with the help of this technique, it can identify the sequence of previously unknown viruses related to those already known, and thus giving us a better understanding of the disease itself. So next, I'll be talking about the applications of PCR. So there are three applications that I would like to talk about today. The first one is the detection of pathogens. The second is cloning. And the last is the identification of identification of a person. So the first is detection of pathogens. So for the detection of pathogens, it is so infectious diseases are caused by pathogenic microorganisms such as bacteria, viruses, parasites, or fungi, and they can be spread directly or indirectly from person to person. The diagnosis of this infection disease has been revolutionized by the development of molecular techniques, mainly with the application of polymerase chain reaction. They are very sensitive and they are used worldwide today by scientists. So how these pathogens can be detected by the help of PCR is that, one slide up, All right. So the method of this detection of pathogen is that first the DNA will be denatured into single strands, and next step is that the extended primers, the DNA will be extended with the primers with the help of TAC DNA polymerase. So the function of TAQ DNA polymerase is in a PSC reaction, PCR reaction is to amplify the DNA for the production of multiple copies of it. And third is to repeat the denature and annealing of primers. Fourth is to extend the primers and tag DNA polymerase. Fifth is to repeat the denaturing and annealing of the primers. And last is to extend the primers with TAQ DNA polymerase. Second is cloning. So in cloning, a PCR-based cloning is about making copies of a piece of DNA, and that at the same time adding restriction sites to the end of that piece of that DNA so it can easily be cloned into plasmid of interest. So how this cloning process works is that it amplifies the DNA fragments of interest with a polymerase using primers that is complementary to the DNA ligase. So the term amplifying is to create, produce multiple copy of itself. And then like for the second procedure is that with each cycle, the resulting DNA is amplified at a very fast rate. The third is the amplified DNA is then joined with the appropriate vector. And this can be done with the help of ligase. And lastly is to insert the plasmid into an E. coli and then spread into an agar plate with the help of an antibiotic for rapid cloning. And last is the detection of identification. So in that, DNA profiling is a technique used by forensic scientists to identify someone based on the DNA profile. PCR can be used as a tool in genetic fingerprint. And for example, like tiny DNA samples like hair, saliva, fingerprints, which is isolated from the crime scene, can be compared with the DNA from suspects or compared with the DNA database. And other than that, PCR can also be used to detect things like to detect biological fathers of a child, if a child is being adopted from another parent. So that's all from me. Thank you.
So next, I'm gonna explain about the genomic library and the applications. So genome is a complete set of genes. It is different from one organism to another. Genomic library refers to genetic information in a genome. So genetic information can be read and interpreted. Construction of genomic library involves recombinant DNA technology. So the objective of Human Genome Project is to create high-resolution genetic and physical maps, and it is also to determine the complete nucleotide sequence of the human genome. So the application of PCR is to amplify DNA sequence to produce enough DNA copies to be analyzed using other techniques. For instance, the DNA may be visualized by the gel electrophoresis, sent for sequencing, or digested with restriction enzymes, and cloned into a plasmid. It also has practical applications in forensics, genetics testing, and diagnostics. For instance, PCR is used to amplify genes associated with genetic disorders from the DNA of patients. Other than that, it also used to test for bacterium or DNA virus in a patient's body. For instance, if the pathogen is present, it may be possible to amplify the regions of its DNA from a blood or tissue sample. That's all from
Thank you, Janaki, for your explanation. Now, I'll be explaining about cDNA. cDNA is a DNA synthesized from a single-stranded DNA template in a reaction which catalyzed by the enzyme reverse transcriptase. So there is three benefits of this cDNA, which is the first one is no introns. What does it mean by no introns is that the eukaryote genes commonly contains introns, which is the non-coding sequences. So these are removed after the mRNA synthesis. So the cDNA contains no introns. So this means that a cDNA copy of a gene can be isolated as a single intron-free fragment. So prokaryotes don't have introns, so this is not a problem if you are working with bacterial genes. So the next benefit is more template. So what does it mean by more template is that there are multiple copies of mRNA for every copy in the genome. So means you will get more copies per cell of the sequence of interest. And the third benefit is less background sequence. So less background sequence means that the sequences that have been expressed as mRNA will be present in a cDNA prep. So there is less background sequence compared to genomic DNA. So this makes it less likely that the primers will bind non-specifically. So when it comes to application, this cDNA is normally used in functional cloning to identify genes on encoded protein functions. And this cloning has been created. You can create like cDNA libraries where it can be used in like UltraScript or UltraScript 2.0 range of products. So it means like collection of DNA sequences that are complementary to the mRNA. Right. That's all. Hello. Hello.
So hi, I will explain about the generating cDNA. So the first one is that we prepare a protein sample. So our protein is a chain of amino acids which is formed from mRNA sequence. So if we know the amino acid sequence of our proteins, then we know our mRNA sequence based on the codon table. So the next one is that we isolate the mRNA from the cell and add the reverse transcriptase to change the mRNA to become a single-stranded complementary DNA or single-stranded cDNA. Then we remove the mRNA by using an enzyme known as RNase enzyme, leaving only the single-stranded cDNA. We then next is we need to convert the single-stranded cDNA into a double-stranded DNA. So we use the help of the enzyme known as the DNA polymerase. So after the process, now we have the double-stranded DNA. So we just need to inject the DNA into a cloning vector, such as the plasmid or a virus. After that, we inject the cloning vector into the bacteria to infect it, and the bacteria will produce lots and lots of the double-stranded cDNA with through a process known as amplification. And once we have lots of this double-stranded DNA, we can sequence that DNA and compile it in a large database or cDNA library online to be easily accessible for anyone. through online. So that's all from me.
All right, thank you so much for that. So I'm gonna start recording.